Hey guys, welcome to your second video involving the cell cycle. So if you recall from the last video, we talked about interphase very exclusively and gave you a brief overview of the cell cycle. Remember, the cell cycle involves interphase, mitosis, and cytokinesis. What I want you to remember is that you're not going to have to memorize these stages. I mean, you'll probably recall them just because we've talked about them quite a bit in class. But what the reason why we're learning about this and the reason why it's so important is because this is what explains how we grow as an organism. This is what explains how we heal ourselves after we get hurt. The process of mitosis and the whole cell cycle is really important in making sure that we grow, we repair our body, and really have all of the tools necessary to ensure our survival as an entire species. So if this process didn't happen, we would not be here. And so I really want to emphasize that while it's not just the memorization, it's also the overall picture. Like, how does this help us as eukaryotes ensure that we survive so that the next generation can continue to populate and continue to ensure the survival of our species? Before we get into detail about mitosis, I want to go ahead and review the information we've talked about with chromosomes. In your notes, you've already added a few things about chromosomes because I told you to add them like on a Post-it. In the new set of notes, I did add that information as well. I just felt like it was probably pretty necessary to write it down again. If you don't want to write it down again, it's okay, but it's just nice to have that information organized into one area. I also just noticed that this area over here is all like it's a white square, and I don't understand why. That's not important. Okay, so let's go ahead and review some of the words that some of the vocabulary that we've already covered. Chromosomes. And so if you look here, this is the typical idea of what we think of when we think of a chromosome, but there's a little more that goes into it, a little more understanding. Yes, a chromosome is one structure, but I want you to understand that X shape that we see right here is actually the result of DNA already being replicated. So when we actually talk about a chromosome, you'll hear the word chromosome and chromatid actually used interchangeably when we look up the structures. So I'm going to show you what I mean here in a second. Let me define chromosome. It's a coiled structure made of DNA. And so all of our DNA is contained on our chromosomes. So chromosomes are the structures that hold the genetic material that tells our body all of the information it needs. Now, we only see chromosomes when we're actually dividing the cell. So that means we only see chromosomes when we are in the process of mitosis. Any other time in the cell cycle, like during interphase or during cytokinesis, we're going to see what's known as chromatin. And so if you look here, this is actually supposed to represent that chromatin-looking structure. It's kind of already been made into chromosomes. But just to recall, chromatin is the uncoiled, unorganized, grainy DNA material. And it doesn't have to be organized, so why would the cell use energy to organize itself if it doesn't have to? I want you to understand that everything in our body, when it's doing something, requires energy. And if we don't have to use energy, we're not going to. It's not efficient. So chromatin is what our DNA is found in when we are not dividing. When the cell is starting to divide or getting ready to divide, it will turn into chromosomes. All right, back to chromosomes. Remember, we see the X structure after we've seen DNA replicate. These two here, and it's named on the picture, these are known as sister chromatids. Each one of these sides, oh man, I got a lot going on here. Let me erase some of this. So each one of these sides, so this side here and this side here, are genetically identical copies of one another. And so when they go to split in half, one side will go to one cell, the other side will go to the other cell. This ensures that both cells that are created get the exact same genetic material. So again, a chromatid is just one half of a chromosome, but each chromatid is genetically identical to one another. So please understand that. A chromatid is genetically identical to its sister. A chromosome is made up of two chromatids. What holds those chromatids together is this button-like structure in the middle known as a centromere. Another thing to keep in mind is you've probably heard the word genes. So genes are pieces of genetic material coding for specific proteins. And so there's a lot of confusion when it comes to chromosomes, DNA, and genes, because it all sounds like the same thing, kind of. We're talking about the genetic material here, but it's actually more of an organizational method. We can think of chromosomes like a big filing cabinet. A filing cabinet, the purpose is to hold information, hold files. So chromosomes are like the filing cabinet. It's the structure. It is just where we contain everything. The actual information inside of that, all of the information inside of that filing cabinet, we can think of as the DNA. And now when we have a filing cabinet, we don't just want to throw everything in there. We want to have it some way organized so we can find things. So the folders that we would have in a filing cabinet are our genes. That is the pieces of information. So for example, inside of my filing cabinet, if I were to go in there and I were to look up cell division, that folder, it would give me all of the information that I have over cell division. If I look up the folder for mitosis, it would give me all of the information for just mitosis. So again, the chromosomes are like the structure. So like the filing cabinet, DNA is all of the information inside of the filing cabinet, and then the folder holding one piece of information are the genes. And hopefully that gives you a better idea of how those three words are used. They're not used interchangeably. There are three different things, but it's more of just an organizational method. Now, when we talk about chromosomes, we are going to spend quite a bit of time talking about human chromosomes, and why that is because human chromosomes involve us. So why wouldn't we talk about us? We've talked about this in class. Let's be cell-centered. So human cells normally have two sets of chromosomes, one set inherited from each parent. That means you get half of the genetic material from mom, half of your genetic material from your dad. 23 chromosomes in each set for a total of 46 per cell. Now, reminder, this is before division. So a normal cell, when it's not dividing, it's just doing its everyday job, has 46 chromosomes there. When we replicate our DNA during the S phase of interphase, we will double that 46 number to ensure both cells each get 46 chromosomes. So please keep that in mind. 46 is a normal cell. 92 chromosomes would be after DNA replication, when we are ready to divide the cell. Chromosomes in a pair are known as homologous chromosomes. Homologous chromosomes means that these two chromosomes, one from mom, one from dad, have the same genetic material on them. For example, they'll both code for hair color. It may be that your dad has a different hair color than your mom, but they both contribute something that gives you your hair color. So you'll see here we have chromosome there. Each are known as chromosome number 1. They are similar in size, and they have the same type of genetic material on them. They are not identical. Homologous chromosomes are not identical, but they have the same types of genetic material. So during DNA synthesis, during the S phase of interphase, both of these chromosomes in number 1, number 2, 3, 4, so on and so forth, both of those will replicate themselves, and that's when we create that X shape. So that X structure. And so then when that information is split in half, we ensure that both of those homologous chromosomes are sent to both cells. And I know it sounds kind of confusing. I will show you what I mean in class with more of a visual, but I want you to understand that these homologous chromosomes are not identical. So it seems like we're using the words chromatid and chromosome interchangeably here. All right, so the second and third parts of the cell cycle are known as mitosis and cytokinesis. Mitosis is where the nucleus divides, and cytokinesis is where the cytoplasm is split. So let's talk about mitosis. There are 4 phases of mitosis. They are known as prophase, metaphase, anaphase, and telophase. And so if you want to remember that, PMAT. And if you want to know the whole cell cycle, you could say IPMAT. So P stands for prophase, and so that's represented here. You'll notice a couple things. There is no nucleus. Remember, the nucleus holds the genetic material of the cell. There's no nucleus here. In prophase, the nucleus is broken down. Chromatin is turned into chromosomes. So you'll see here we already have those X-shaped structures. A reminder that these cells, these two things here, are known as sister chromatids. They are genetically identical pieces of one another, and they will get split in half. And so this is two different chromosomes here. And also in prophase, we see that the centrioles will move to the sides. And if you remember a few months ago when we talked about animal cells are the only cells that have centrioles. It's not really represented here, but we would have them kind of like on the sides here. So in prophase, we will start to see centrioles move to the opposite sides of one another. And sometimes you'll hear the opposite side known as a pole. And so you'll see that in some of your notes. In metaphase, metaphase starts with an M, and so does the word middle. These chromosomes will then start to line up in the middle. That way we can ensure that both sides get split up. In anaphase, those chromosomes, the chromatids get pulled apart. So you'll see here that this chromatid goes to this side and this chromatid goes to that side. Same with that magenta looking color. So the purple and the magenta both get split. In telophase, it is the exact opposite of prophase. So in telophase, we're going to start to see the nucleus start to rebuild itself. We'll start to see those chromatids moving away from the center, moving away from the sides. And then we'll start to see those chromosomes start to break back down into that grainy material. Not completely, but you'll start to see that process of turning back into chromatin. But the only thing is that we have split the nucleus. We have two nuclei, but the cell is still attached in the center just by you could think of it as just like a skin material, gel-like material. And then cytokinesis happens, and this is the third part of the cell cycle. In cytokinesis, we see the splitting of the cytoplasm, making two brand new cells with their own plasma membrane surrounding them, and then they start to grow, going through the G1 phase, and then eventually G2. So that continuous cell cycle happens right after cytokinesis. Now, I don't feel like I've given you brand new information because we spent quite a bit of time talking about this already in class, but please make sure that you're writing your notes, you're asking questions, you're following along. This is going to be really important as we go into the next new school year, not new school year, but the new year. So please keep up with all the information in class. If you have any questions, of course, ask me on Edpuzzle.